Good evening, friends. Hi, good evening, all of you. Hi, my name is Srikant Singh, and I'm the biology educator. I'm the biology educator. So today, beta, I'm going to talk to you about the topic in front of you: cell cycle and cell division. Let's start. The chapter name is cell cycle, cell cycle, and cell division, and cell division. Interesting chapter. Now, question is arise, beta, that why cell is going to be required for the cell division. Hmm? Why cell require division? Why cells require or need division? Answer is first is सेल ग्रोथ. जाहिर सी बात है, कभी किसी ऑर्गेनिज्म को ग्रोथ करना हो तो ग्रोथ के लिए डिवीजन इज वेरी इंपॉर्टेंट. इसके बाद नेक्स्ट उंड उंड हीलिंग. अगर कोई चोट लगती है तो उसमें भी सेल डिवीजन इज वेरी इंपॉर्टेंट. नेक्स्ट नेक्स्ट इज wound healing, cell growth, wound healing, cell repair. If any cells is going to be damaged, should be repaired with cell division. Fine. Fourth, for for development for development for development of of the the organism body shape. For the development of organism body shape. This is a few point which cells division is required. Now, there is a two type of cells, beta. Always remember, there is two type of cells. Hashtag two type of cells. Two type of cell. Two type of cell. First is somatic cell. Somatic cell is a cells which doesn't carry genetic material from one generation to another generations. It is also called body cells. Our whole organism body is made by somatic cell. Is it clear, students? Now, second cell is germ cells. Germ cells are always present in the gonads. Like in the gamete, the germ cells is present, which is responsible to carry genetic information from one generation to another generation. Suppose that if any persons have gym personality, it doesn't mean that its son also having the gym personality. Why? because it is a somatic cell. It's for practicing, for doing hard working, it adapts its body shape. But in the case of germ cells, it is the genetic, what is the quality have the parents cells has to be transferred from to the next generation to the daughter cells. So germ cells is the cells which can carry the genetic information from one generation to another cells. In the somatic cells, there is two type of cell cycle is running for division. His name is one is mitosis. Always remember, in the somatic cells, mitosis cell division is occurring. It is called differentiated cell. It is called equational division. It is called beta. Always remember. Mitosis is also called equational division. Mitosis is always called equational division. Why we call equational division, students? See, notice the word, what I'm writing. Equational division. Equational division. It means parent cells, suppose that parent cells having the 46 chromosome. 46 chromosome. After the division, after division, two daughter cells is going to be formed. Two daughter cells is going to be formed. But it also having the 46 chromosome in their body. So due to this reason only, we call mitosis is called equational division. And in the germ cells beta, it is reductional division. Let's start. See, it is the parent cells. It is the P stand for parent cells. Okay. So in this germ cells, mitosis process is not going to be occur. It have in these cells meiosis process is occurring. Meiosis process. In the meiosis, there is meiosis process. It is also called reductional division. It is also called reductional division beta. It is also called reductional division. Suppose it parent cells. So meiosis, meiosis is meiosis has completed into two phase. Meiosis is going to be completed into two phase. Meiosis phase is going to be completed beta in two phase. Meiosis phase is going to be completed into two phase. Which one? Let's take a look. Meiosis 1. Meiosis 1. Meiosis 1 beta, always remember it is the reductional division. Which type of division it has? Reduction division. Means in this phase, chromosome number is going to be half. Reductional division. Reductional division. Chromosome number is going to be half. But if you talk about the meiosis 2, if you talk about the meiosis 2, then this phase is similar to the mitosis process. So it is the equational division. It is the equational division. Equational division. Due to this reason only, in the first meiosis division one, meiosis one divisions, chromosome number is going to be half. The parent cells is going to be form two daughter cells to form two daughter cells. and these both daughter cells having the 23 chromosome, and this three both daughter cells is going to be participated in the meiosis 2. So this way, meiosis 2 form the four cells having the chromosome number 23. This one is called, and it is the, so this is the condition. Now we can define. Now let's we understand about the, now let's we understand the cell cycle. For cell division, it is the it is the it is the biological process beta. Always remember, it is the biological process. It is the biological process in sequence event. It is the biological process in sequence order. In sequence order. In sequence order. In a sequence order to form daughter cells. To form daughter cells. To form daughter cells from parent cells. To form daughter cells. Now, suppose that this is the, it is the cell division. Is it okay, beta? Cell division is divided into, has to be into two part. Process is going to be completed in two step. First is interface. Interface. क्या होता है interface, बेटा? Interface. So interface is the preparatory phase. Means cell खुद को prepare करता है कि when cell has to be divided, and while division cell is ready or not. That is also important. So in interface, it is also called preparatory phase. Preparatory phase. Preparatory phase. Preparatory phase. A second is M phase. M stand for mitosis process beta. M stand for mitosis. M stand for mitosis. Cell has to be there. Now, interface is divided into few substep, name is G1 phase. G1 phase. So beta G1 phase, in this phase, chromatin, the thread-like structure is called chromatin, which is going to be start the condense. It's going to be start in the condense. Okay, the thread-like structure chromatin is going to be start the condense in G1 phase. Why? Now, in this phase only, G1 phase, chromatin start condense. Chromatin start condense. Tubulin protein is going to be formed in G1 phase. Tubulin protein is going to be formed tubulin protein enzyme for replication, enzyme like helicase enzyme, beta helicase enzyme, topoisomerase enzyme, primase enzyme, whatever the enzyme and protein required for the cell division, histone protein, all thing is going to be manufacturing in the G1 phase only, and also duplicate of the centriole, and also has to be duplications of the organelle like endoplasmic reticulum, Golgi apparatus is going to be duplicate. Mitochondria is going to be duplicated in the organelle of duplications. Duplicate, duplicate organelle, organelle. This is the G1 phase beta. Now, in the next step is S phase. S phase. S phase is the synthesis phase beta. Always remember, S phase is the synthesis phase. In this phase, replication process start. Replication process done. In G1 phase, chromosome number is 2n is equal to 4. So, in the replication, when it is division replication process occur, then the quantity of the cell volume also is going to be double. So 2n is equal to 4, 2c is going to be converted into 4c. In here, till 2c is the volume of the chromosome. Now, in the replication process is occurring, histone protein is formed. After that, replication process, S phase, G2 phase is going to be formed. In G2 phase, rest of the organelle, rest of the organelle, rest of the organelle, rest of the organelle is duplicated, is duplicated, is duplicated, is duplicated. So this way, and one is G0 phase. G0 phase. G0 phase is the quiescent phase. It is the resting phase. In this phase, when the cell is going to be damaged and it is not capable to divide, then it's going for before entering in the G1 phase. Before entering in the G1 phase, the cells goes to the G0 phase for repair, and then come back to enter in the G1 phase for the division. So it is a G0 phase. In the NCT, it is the preparative phase. It is a preparative phase I have already taught you. Now, the rest phase is name is M phase. M phase is mitosis. Mitosis process is in two phase. Name is during step in two step. Is completed into two step. First is karyokinesis beta. Always remember karyokinesis. Karyokinesis. Karyokinesis is going to be completed in few steps. Name is prophase. Few step name is prophase. And then prophase, metaphase, and anaphase. 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 And then telophase. And then telophase. So this way, telophase. So this way karyokinesis is completed. Karyo means always remember, karyo means it means beta nucleus. It means nucleus. In nucleus. Kinesis means always remember division. It is always so karyo means division of nucleus is called karyokinesis, and second step is cytokinesis. After nucleus division, cytokinesis is going to be occur. Division of cytoplasm is called. So this way, mm, cytokinesis. Now, if we talk about that, so this way, why cells require division, cell growth, wound healing, is and for the cell repairing, it's required cell division. For development of the organism body shape also required cell division. And there is a two type of cells. One is somatic cells and the second is germ cells. Somatic cells having the body cells. In these cells, always the mitosis cycle, cell cycle is going to be occur. Mitosis cell cycle is going to be occur beta. And mitosis is also called equational division. Why it is called equational division? Because parent cells having the chromosome number 46, but after division and forming the daughter cells also having the chromosome number same 46 and 46. So that's why mitosis is the cells which doesn't carry the genetic information from one cell to another cells, but germ cells you take in these cells, the always it occur in the gamete, in the gonads, to carry the genetic information from one generation to another. So this way, it is the mitosis process. This way, the mitosis process is going to be completed. Duplicate, genotype, and telophase, canokinesis, and then telophase. After that, inherit and cytokinesis. And what about the meiosis process? Karyokinesis. In meiosis, in meiosis 1, in meiosis 1, in meiosis 1 beta, always remember, it is divided into two phases, meiosis 1 and meiosis 2. So it is a reductional division prophase 1. Prophase 1 is very big. So that's why it's leptotin. Leptotin. Leptotin, zygotin, zygotin, leptotin, zygotin, packaging, and diplotin, and diplotin, diplotin. So this way, the prophase one is longer in meiosis one. After that, meiosis, prophase one, metaphase one. After that, metaphase one, metaphase one, metaphase one. And after that, metaphase where anaphase one, simple. Just go through the step of the mitosis. Metaphase one, anaphase 1, anaphase 1, anaphase 1, meiosis 1, and second phase is meiosis 2, meiosis 2, meiosis 2 is just similar in the like mitosis. One is sorry, prophase 2, prophase 2, prophase 2, metaphase 2, metaphase 2, metaphase 2, then anaphase 2, anaphase 2, anaphase 2, and then telophase 2, and then telophase 2 beta, then telophase 2. Up now, this is the complete cell division explanations classification I'll show you beta. Now go for it. Suppose that next page. Suppose that it is a chromosome. It have P arm, and it is the Q arm. P arm is the short arm. P arm is short arm. Chromosome having the short arm and long arm. Long arm. Always remember beta. And between these two arms is connected by centromere in the chromosome. Centromere is going to be connected with centromere. So this way, cell division process has occurred. Now, if we talk about the orally, when we talk about the orally, in the mitosis process, first step is karyokinesis. In the karyokinesis, prophase phase, in this phase, prophase phase, first step is prophase phase. Suppose this is the diagram. In this phase, the chromatin is going to be start condense. As you seen that starting time, the prophase phase have the chromatin. It is in long in length and thin, because now it is going to be starting to condense in the prophase phase. Prophase phase having the chromatin, and it is going to be condense. But in this phase only, and this phase only, nuclear envelope is going to be disappear. Nuclear envelope is going to be disappear. Golgi apparatus is going to be disappear, endoplasmic reticulum is going to be disappear, mostly organally, and nuclear membrane is going to be disappear because the cell division is going to be started. And end of the prophase, 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 centriole is going to be duplicated. Centriole is going to be duplicated. Here it is, the centriole. This is the phase of prophase. After prophase, it is the metaphase. Metaphase. In the metaphase phase, the centriole is already duplicated, so it is the centriole start going to be moving. Start moving to metaphase phase. And chromosome number is already is contains. In metaphase, the chromosome is going to be arranged in the centripetal plate. The chromosome is going to be arranged in the centripetal plate. Centripetal plate. As this spindle fiber is going to be attached to the chromosome at the site of attachment. Name is काइनेटोकोर बीटा. काइनेटोकोर कहाँ ये अटैच होता है क्रोमोसोम पे? काइनेटोकोर पे अटैच होता है. काइनेटोकोर बिटवीन द क्रोमोसोम आर्म्स हैविंग द सेंट्रोमियर. और मेटाफेज में आपका काइनेटोकोर में स्पिंडल फाइबर जो है बोथ पोल्स से आपका स्पिंडल फाइबर किस चीज का बना हुआ है? किस प्रोटीन का बना हुआ है? ट्यूबुलिन प्रोटीन का बना हुआ है. और ये डायरेक्टली अटैच किस पे करता है? काइनेटोकोर पर. अब अटैच होने के बाद इस तरह से ये सेंट्रीपिटल प्लेट में अरेंज हो जाते हैं. नाउ नेक्स्ट स्टेप इज एनाफेज बेटा. एनाफेज स्टेज. एनाफेज में स्टेज में क्या होगा बेटा? इसमें एक एंजाइम आएगा जिसका नाम है सेपरेज एंजाइम. सेपरेज एंजाइम. अब ये एंजाइम क्या करेगा? सेंट्रोमियर को ब्रेक डाउन करेगा. जाहिर सी बात है कि जब तक सेंट्रोमियर ब्रेक डाउन नहीं होगा तो ये ऑपोजिट पोल की तरफ मूव ऑन नहीं करेंगे. सो इस तरह से हम देखते हैं कि एनाफेज के टाइम पीरियड पर मेटाफेज एनाफेज के टाइम पीरियड पर ये एनाफेज के टाइम पीरियड पर ये ऑपोजिट पोल की तरफ मूव ऑन करना स्टार्ट हो जाते हैं. एनाफेज after that it is the telophase. In telophase अब ये क्रोमोसोम जो है क्रोमोसोम जो है अपने अपने पोल पर ये रीच कर जाते हैं. रीच करने के बाद इनके ऊपर में बाहर से अपना जो न्यूक्लियर मेम्ब्रेन इज गोइंग टू बी डिसअपियर वो फिर से अपीयर हो जाते हैं. इस तरह से कैरियोकाइनेसिस इज गोइंग टू बी कंप्लीटेड. अब इसके बाद स्टेप आता है आपका बेटा साइटोकाइनेसिस. साइटोकाइनेसिस में होता क्या है कि प्लांट सेल्स में गोल्जी एपरेटस का डिसोसिएशन होता है और फिर वो ब्रेकडाउन होने के बाद वो सेल प्लेट जो है वो एक फॉर्म करते हैं. अब सेंट्रल पीटल एरिया में ये प्लेट फॉर्म होने के बाद फिर ये फॉर्म होने के बाद सेंट्रीफ्यूगल फोर्स अप्लाई होता है. सेंट्रीफ्यूगल फोर्स हो यानी अवे फ्रॉम द सेंटर एंड दिस वे two daughter cells is going to be formed in the plant. ये हुआ प्लांट सेल्स की बात हुई. प्लांट सेल्स में आपका सेंटीपीटल प्लेटफॉर्म होता है, और फिर वो सेंट्रीफ्यूगल फोर्स अप्लाई होने के बाद ये पेरेंट सेल जो है डिवाइड होता है टू डॉटर सेल्स में डिवाइड हो जाते हैं. आफ्टर दैट एनिमल सेल्स एनिमल सेल्स एनिमल सेल्स बेटा इज गोइंग टू बी, तो ये एक आपका पेरेंट सेल्स है. ये समझो कि ये फोरो लाइक स्ट्रक्चर फॉर्म हुआ. एक फोरो लाइक स्ट्रक्चर, अब ये धीरे धीरे जो है सेंट्रीपिटल की तरफ और इंक्रीज होते रहा, और फाइनली ये डिवाइड होता है, और डिवाइड होने के बाद फिर ये टू डॉटर टू डॉटर सेल्स में, इस तरह से आपका माइटोसिस प्रोसेस स्टार्ट होता है. प्रोसेस इज गोइंग टू बी कंप्लीटेड. अब अब so this is the overview I have explained in front of you about the cell cycle and cell divisions classification I have taught, and the mitosis I have tried to explain it in short. So thank you so much, students. So giving you a valuable time. Thank you. Stay blessed.